Thank you for being here. We have a full house. I know it's exciting. You are in for a treat today. This is our final talk of the winter spring. And we're so lucky to have real live marine biologist Nancy Treneman. Hi. Yeah. From Oregon, who is going to teach us about all kinds of interesting critters.
Right. So I'll just do a self-intro. I grew up in McMinnville, Oregon. Anybody from McMinnville? Salem? Salem's more boring than McMinnville. So, and I used to come down here through the OMSI, which is the Oregon Museum of Science and History. We had a marine biology institute there in Cape Arago. And I went every year from 5th grade to high school and did Tidepool out of Cape Arago and down through Cape Blanco all the way to Gold Beach. And the reason I'm in Gold Beach now is because when I got a chance to move, I said, I love those beaches. So that's why I moved down there originally. I taught high school for 30 years over different schools in Oregon. Gold Beach for the last 10 years of my career there. And then for the last 10, I've been courtesy faculty at the Oregon Institute of Marine Biology, which is a University of Oregon campus. How many people have been to our wonderful aquarium? Yeah, the CMLC. You see those beautiful whale skeletons? I helped build those. Yeah. And if you get a chance, make sure you go because me and the team just finished a infant orca skeleton, which is probably one of the only ones in existence. So very cool. I do skeletons, and I am a marine biologist. I work with a lot of different marine vertebrates and seaweeds. And today I just wanted to share with you some of the really unusual, but odd and tiny creatures that I see out in the Tidepool. And you'll be able to see a lot of them yourself if you are walking the beach and walking rocks, especially if you go up to Cape Arago or along the rack line here in Bandon. You got lots of cool stuff. So we're going to cover. We have some kind of echo. Right. So I'm going to just take a second to X that out. How's that? Perfect. I hope I didn't destroy the. Nope. You're okay. Fine. So anybody recognize any of these three organisms? There's David. David, Diana. Right. Dave probably helped me pick some stuff. Diana. So this is a segmented worm. Like your worms. But in the marine environment, the segmented worms are called polychaetes because they have lots of little bristles. This is a porcelain crab. They're very tiny, about the size of your thumbnail. This is a pteropod. Pteropods are snails that swim in the ocean. They don't crawl around anymore. They have very reduced shells inside this really unusual body. And these wash up along the rack. We're also going to hit sea cucumbers. I just didn't have enough room in there to do it. So you go out to the rocks and you see beautiful environment like this on a low tide. This is that, looks like that's at Harris Beach down where I live down in South. And this is the environment that I basically feel most home in. You have lots of eelgrass here, which is a flowering plant. But my favorite flora, of course, is the seaweeds, which I am teaching a class right now at Oregon Institute of Marine Biology. So when you hit the rocks, you're probably familiar with this kind of seed. And you probably recognize a lot of these organisms. These are your standard Oregon marine invertebrates that you see in all of the different advertisements. We have Physastrocracius, which is the orange sea star. It comes in three colors, though: purple, brown, and orange. You see the green sea anemone, the big one, Anthopleuria and Xanthogrammica. But you also, and you see, and this picture, not a lot of seaweed, all the little tiny anemones up above are a different kind of anemone. They have little pink tentacles, Elegantissima. The California mussel, which some of us like to eat. And if you hit the seaweed, if you go into a little lower tide zone, especially if you go to the rocky intertidal as opposed to the seamounts that are in the sand, there's some really amazing kelp in our ecosystem, which is basically responsible for our ecosystem. And if you turn over rocks, there's an incredible number of encrusting species that live under and around rocks. And that is something that I spend a lot of time looking at rocks like this. In that rock, on that rock, you have a sea urchin and a yellow, purple sponge. Under the rock, you have this lovely polychaetes. These are filter feeders, tunicates, which are, they, bizarrely enough, actually the most closely related invertebrate to us, to vertebrates. You have nudibranchs. How can you not love nudibranchs? And chitons. How many people have seen some of this stuff? Cool. So here are the scientific names of these. And I can turn over a rock and find, if I worked at it, 20 species on that rock. And they're all fairly small. But the more you look, the more you see. And the smaller you look, the more you see. So one of the things that's really amazing about marine invertebrates is that if you start looking small, then you see a lot more. So the pysasters and mussels, you see, I mean, those are all cool. I love those guys. But the unusual, odd ones that we don't hear much about are usually pretty tiny. So the segmented worms. Polychaete means many bristles. And the polychaetes are marine. They live from the deep sea all the way up to the intertidal zone. The deep sea, sometimes the deep sea ooze is just coated with polychaetes. They're very abundant and diverse. They can be predators. Some of them have jaws that literally, if you bite, will bite you and it really hurts. They can eat little bits of food that's called detritus, little broken up pieces of seaweed and meat and stuff in the water and in the sand. They can be parasites. They can be filter feeders, which means they take the water and they filter out the plankton and little zooplanktons. This is one of the predatory ones. These can be like this big. When they try to get out of the dish, I feel like they're attacking me. Oh, by the way, they are the inspiration for the sandbar. You do, because Frank Herbert came to Florence and saw those dunes, and he wrote that book, and he found out about these odd polychaetes. So we are so famous. No one knows that, but we do. This is a filter feeder from our mudflats. They're big Nicula, really beautiful, fantastic kind of bloody gills on the side. These are fantastic. These are the stipulas. They secrete this calcareous tube, and they have these flower-like appendages that filter the water. They're just gorgeous. And these are the scale worms, which in our environment can be maybe an inch or two long, but there are really large scale worms in the tropics that are just outrageous colors. We have some really beautiful ones as well. So you turn over the rock, and in generally boulders which are in a sandy substrate, you're going to find these terabellids, which are the spaghetti worms. And they build a tube under the rock out of chitin and the pebbles and sand, and they live inside that tube. And you can see a few of them here. You have to get good at recognizing them. All right. And it always bothers me when I turn over a rock and there they are, half out of their tube, very upset. So this is what one looks like. They're just amazingly beautiful. You can see the segments here. It also changes as it goes down. These are the tentacles that collect the detritus from the sand. And these are the gills right here. So the tentacles are sticky. They pick up little bits of food, and then they literally are covered with mucus. And then the worm puts it in its mouth and kind of, you know, takes it off, slurps it off. Teenagers would love that. Red gills. So if you look here close up, you can see the little gills. They're full of hemoglobin. And it turns out that the blood in these polychaetes has some really unusual properties. And people are spending a lot of time studying them when it comes to oxygen binding and the chemical reactions in them. So here you can see the what's the word, Nancy? Segments up close. And I don't know if you can see, but there's little tiny bristles. There's little tiny paddle-like things on each segment. And those are called parapodia. And the structure of the bristles and the placement of them helps you identify what species as well as what the head looks like. Identifying polychaetes is not for the faint-hearted. Very few people actually do it anymore, which is sad. They're outrageous. So as you get closer, you can see even more. So the closer you get, the more fantastic these very simple organisms look like, which at first you might say, ooh, it's a worm. No, it's fantastic evolutionary production that produced these amazing animals. So they produce all kinds of cool chemicals, it turns out. This one here that is specific to our local species is called Philippamide, after Philippus, which is its scientific name. And amazingly enough, this thing has a real effective action against leukemia. So polychaetes have become extremely famous in the biomedical world for all of their chemicals, which we'll get back to a little bit. But I thought I would show you just kind of one of them in action here. So this is, well, it doesn't show up too much on the Zoom, but there you can kind of see the action of the, it's not so, it's smoother on my screen than it is yours. But you can see the action of the tentacles, and they spread way out. There's one in Hawaii, which actually terrified me, that has tentacles about 1 meter long. And I was walking around the Tidepools, which is all lava, and I sat down and I looked over and there was this giant white tentacles, literally as long as my arm, coming out of this hole. And I thought, oh my God, you know, it's gonna eat me or something. And I had to go look it up and talk to a bunch of colleagues, and it turns out it's called the Medusa worm. And you never see the body because it's inside the lava, which technically is insanely hard to get in. But you sure see those tentacles. So the Medusa worm is pretty neat, very famous in Hawaii for terrifying visitors like me. So the Philippus crispus, which is the one we just saw, it makes a burrow, and it spreads out its tentacles, and it produces all kinds of chemicals, which probably are good at first of all, deterring predators, but also deterring competitors. You know, the other worms don't tend to settle where within the zone that this chemical is deposited in. So this chemical has a function for the animal. Here's another one, which I hope. This is my favorite one because he's colored. Look at that great green. I mean, wow. So fantastic yellow tentacles with that great green body. Can't remember the scientific name of this one. And I find them together all the time. The pink one, the brown, there's a couple different colors. There's one that's really outrageously orange, but it's really, really fragile. And every time I pick it up, it falls apart. So I leave it alone. I see it's like touching because I don't want to hurt it.
How much is that magnified?
Oh, let's see here. So you see the rim of the dish that it's in? Yeah. That is you know, you go to the store and you buy those stacked bead dishes. They're about this big around. That's what I take out in pools to do this. And so it's probably, it's all squished up, but it's probably it's all squished up. It's about as long as my upper thumb joint. But when you stretch it out, it's going to be a couple inches, maybe 4. And they can, when they're happy and they're in their tube, the tubes can be 5 or 6 inches long. But they're usually curved and stuff. Yeah, question back.
What's the green?
The green is a pigment in the worm, and I don't really know the reason beyond coloration because these are all buried. There's a couple hypotheses I can give to you. One is probably it produces some chemical that is green, and therefore that's part of its nature. It might also process a food which technically has chloroplast, has green in it, so maybe it prefers seaweed bits. You know, no one knows.
It's not sequestering algae or anything.
No, because it lives under the rocks. But it might eat seaweed and be green because of that. What the great thing about these organisms, all these questions you're asking, most people don't know the answers to because no one's done research. So millions of PhD projects out there. When I take the kids out, great PhD project, you know, they've been 4 years figuring that one out, and add to the wonderful body of knowledge we have about our marine vertebrates. So this is an example of what the biomedical community does with our polychaetes. We've got all kinds of applications to humans. We have anticoagulant drugs, pesticides for your skin. You've got from the skin you get chromophores and antimicrobials. From the whole body you get anti-cancer compounds. The mucins are antifouls because other organisms don't settle on it. It's pretty amazing. So one of the things that's interesting about when I talk about things like seaweed and polychaetes, you often get a question like, well, what good is it to me? You know, and of course, all of these organisms have ecosystem services. So the air you breathe, the land you walk on, the ecosystem you depend on, depends on all these organisms. And our ocean, without our ocean, the Earth would of course be probably pretty lifeless, right? And these organisms are all part of that ecosystem. An ecosystem is like a giant pile of bricks that are all balanced on each other. And you can take a few out, right? But which brick do you take out that then the whole thing collapses? We don't know which brick counts. In fact, probably most of them do. So it's really to our advantage to not mess with success. To keep our organisms, keep common things common, keep rare things as common as we can, but realize the value of an organism is not based on this in the end. It's based on our ability to survive and breathe and eat on the earth, along with everything else. So my sister Mae, who's a nurse, she's pretty funny. They're teaching seaweeds at OIG right now. And she asked me what I was teaching. She lives in California. And I said to her, well, I'm teaching a class in seaweeds. She said, oh, wow. The whole class on just seaweeds? I thought people spend their lives studying seaweeds and haven't finished up. So that's kind of the perception sometimes of these organisms. We want to think beyond that. So let's get into arthropods. We have lots of really cool arthropods here in our intertidal zone. And we're going to focus on the crabs, which are, first of all, insane and cute, but also they have such an attitude, you know? The crabs are always, no matter how small they're all, they want to kill you. And you say, but you only weigh 0.05 grams, and the hermit crab just said, fine, just give it to me, give it to me. I'm gonna hit you. Like, no, I'll just put you back in the tide pool and you can beat each other up. So the Cryptolithoides turtle crab is really, really, I don't see them very often. They're always in a crack in the low intertidal zones, but this is the carapace, and underneath are all the feet. There's also a real similar one called the turtle crab. This is a little tiny Oregonensis that looks a lot like, it's like the pygmy rock crab. So these are sea urchin spines here. You can see how tiny it is. And you always find it in a hole. They like to be in the holes, and you shine your light in there. There's one, and he raises his claws. This is the hermits, absolutely one of my favorites because they will beat each other up just for fun. They pull each other out of their shells. They grab each other and drag each other around for no apparent reason. And then like if you have an empty shell and bowl, I've only seen this a couple times because it doesn't happen very often. So you have an empty shell and bowl that fits one of them, and you have say 10 hermits in there. One of them will decide that's a better shell, and will change to that shell. The next one will change to its shell. The next one will change to its shell. And they all trade. And then what's bizarre is some of those are backwards again. Say, why? Why are you wasting this energy? No, it's all being a hermit. This is the thick clawed crab, lives strictly in rock cavities, and usually if you shine your light in here, that's all you see is a big giant claw. They're just wonderful organisms. This is a crab. Can you see the crab? All that white stuff is not part of the crab. That's decorated. This is the decorator crab. It's covered itself with hydrozoans and bryozoans, which are little tiny encrusting organisms. And when I saw this, it was sitting in a bryozoan patch. And the only reason I knew it was a crab is it moved. So I put it on some red algae so you could really see. And this thing was about, half an inch. Decorator crab, lovely. We're going to talk about this guy. This is the thick clawed porcelain crab, Pachycheles rudis. Pachy, thick, Cheles claw, rudis, red. It has little red mouth parts. And the porcelain crabs, if you turn over a rock in the intertidal zone, most of the time there's about 1,000 under there. They all scramble for shelter, and if you pick them up, they drop their claw, and their claws wiggling, and they run off. So, and they're very, very thin and kind of flattened.
Wait, they drop their claw?
Oh yeah, they'll drop their claw right away because the predator eats the claw, and they're off and food. So some crabs do that more than others. Like if a crab loses a claw, it'll grow one back on the next molt. It'll be tiny, but it'll eventually get bigger. But some the porcelains will drop it you just pick up the crab a lot of times, and it was ready. So it's a predator defense mechanism, but you always feel sad about it. Bummer. Didn't mean to do that to you, young lady. So crabs belong to the group of the crabs are decapods. Deca means hand. Pod means foot, right? Podiatrist. So decapods have 10 feet. And if you count, next time you get crab in the store, you count them, you'll see 10, if you include the front claws, which are part of the system. But in the porcelain crabs and the anamurans, which are the kinds of crabs whose antennae are outside their eyes, they don't have very visible feet. So there's one. The cheliped, which is the claw, the big claw, chele, claw, all right? Defense, sometimes they just do it to block stuff, all right? The boys wave it sometimes at the girl. She usually much. The second antenna is out here, outside the eye in the anamurans. In the brachyurans, like the Dungeness crab, it'll be inside the eyes. That's one of the good tells. Then here's the maxillas are under here. These crabs are filter feeders. They do not pick bits of matter, kill things and eat them. They have these big fringes on their maxilliped, and they'll wave it in the water, and they filter. They don't eat like regular, like what you think of as crabs. A lot of crabs are filter feeders, especially the ones stuck in holes, like this one. And then here's the fifth leg down here. So if you look, 1, 2, 3, 4, right? Where's the fifth leg? It's tucked under here, and it's used to clean the carapace. It's a little cleaner leg. And it's really fun. If they get really comfortable in your dish, they'll start doing that sometimes. The crab is simply a shrimp whose tail is folded up under the body, right? The girl uses that tail to brood her eggs. So if you've ever found a red rock crab and their title's on turn it over, it's a girl, a lot of them are, you see huge piles of eggs underneath that tail flap. Millions of them. So the fifth leg here is used for grooming and cleaning the gills.
I have a question.
Yeah.
You just said they're shrimp. Like
they're like a shrimp,
right?
But shrimp are also crustaceans, but
Okay.
they're not brachyurans. So crustaceans are huge. There's a huge bunch of different kinds, and there's shrimp, crabs, amphipods, all kinds of other ones. And if you look at a shrimp, you're gonna see that long back end.
Yeah.
If you fold it under the rest of the big carapace, that's essentially what a crab is. They are crustaceans who have folded their abdominal sections back under their body.
Are they that closely related?
Well, they're in different groups. I mean, evolutionary, yeah, they evolved from a common ancestor. Yeah. I couldn't tell you the actual period of time span. So here's your porcelain crabs. The second antenna is outside the eye. The back legs are small and tucked under the abdomen. This is the one we're looking at, or Pectacilius rudis. Thick-clawed crab. Notice one claw is much thicker than the other. Yeah, that's something to pay attention to. And you generally find it in holes. So when I turn over a rock, there's a lot of them have little cavities in them, and you'll see these thick-clawed crabs inside the cavity. They also live in cavities and kelp fats and all kinds of cavities, wood too. So weirdly enough, there's left-handed and right-handed. So you have your lefty, you have your righty. They're the same species, it's like humans, right and left-handed. So here's a little, and of course this isn't going to be as good on. So this is one I picked up a holdfast and pulled it open, and there was one of them living inside its cavity. And so here they're upset because of course I've opened up its little house. He's trying to get out. It looks a lot better on my screen, but that's okay. But we got to improve our bandwidth here to get this moving faster. So then, of course, escape from the giant human predator, who's destroyed my home. And he decides, I think we should just go back in. So that's just a little vision of what you see. It's found in cavities, and it's always found together with members of the opposite sex. So you have heterosexual pairs inside these cavities. And you can't tell that there's two in that cavity. And this is another crab. That's a decorator crab sitting on top of it. But there's two little thick-clawed crabs in that cavity. It turns out that, in general, there are always different sexes. And they grow so big that they sometimes can't get out of their cavity. They're stuck in there forever. All right. So you better like your partner, right? There's no sexual dimorphism. So what does that mean? Dimorphism. Dimorph is shape. So humans have sexual dimorphism. Males look different than females. But in this particular species, they look the same, right? The tail. The male has a very narrow triangular abdominal plate. So the tail, which is up underneath, think about it, my hands being my legs folding up, right? The male's is triangular and small. The girl's is very wide and round because she keeps her eggs. That's where she broods her eggs. And so if you look at the Dungeness, the same thing is true. How many people have seen that in the Dungeness crab? If you crab her, you throw the girls back in, right? Good. That's why we have such great crab industries. We throw the girls back in. If we didn't, we would be in bad shape. So decapods, the female crab is not that thrilled with the whole process. And if the male gets close to her when she's all buff and her shell is hard, she'll pull his legs off and throw away. It doesn't work too well. So he has to wait until she's molted. Remember, they can't expand like us. Their skin's not stretchy. Their hard outer covering doesn't stretch. How do you get bigger? You have to pull out of that covering. It's actually a very dangerous process. What they do is they, underneath the old covering, the hard one, a new one is forming. And the old one, as it matures, the old one will come loose. And the crab, the back end, the kind of disintegrates. And the crab has to pull out of that. And they can suffocate during this process because they actually uncover their gill coverings and their eye coverings are part of their skeleton. So they pull out and they're really soft. And then they suck in a lot of water and expand that outer covering. And then they can go into it. Right? So they're soft during that period of time. And if you've anybody ever felt that, had a crab who is just molten? Well, that's when the guy can nail girl because she doesn't have any hard claws, and he grabbed her, and he might sit there and this might go on for like a day. You know, he gets a hold of her, and he sometimes, they'll sense when they're molting, and the male will, so yeah, exactly. And then once the mating occurs, the female broods the eggs. So here's the boy, poor young lady here. And they're stuck like that. I photographed these crabs at 8:30 in the morning, and I went back at 10:30. It was still going on. So, that's a red crop. And so there's the female red rock crab brooding eggs. Notice that they really expand way beyond her tail plate. You find a lot of these in our intertidal zone. And here is the female of our thick clawed crab. And you can see you can't really see as well, but she's brooding eggs right there. All right. She produces 3 broods a year. That's a lot of work, all right? A lot of energy. So monogamy, which means you stay with the same mate all your life, is very rare in invertebrates. But it seems that these particular species do that. And, if you're permanently stuck in this hole, there is a interesting thing where why would you be monogamous at all? So you're going to be monogamous if you were stuck together. You're going to be monogamous if mates are rare. Then you find your mate and you keep them with you all the time, right? So low abundance, and this is an advantage to the male because then he has to go off and search for girls. Otherwise. Now the question you can ask is, do they match up according to handedness? Do lefties always go with lefties or vice versa? So some interesting questions here. How do they form? Do they choose their mate, or do they just happenstance end up in the same hole together and boot one out if they're the same sex, that kind of thing? Will they recognize strange crabs that are not their mate? And will they aggress them and keep them out of their hole? There are all kinds of questions you can ask about this system. Do they recognize their mate if you take it away? Right? Let's find out. Now, the really great study was done at some point to figure this out. So what happens is that the female is occupying a hole, and the male comes in, and she'll grab his chelate, and she'll grab his claws with her big claw, and she'll push down. And the researchers say you can actually hear this crunch. She doesn't take it off or anything, but she pinches it. Well, if the male's bigger than her, he tends to fight back, and she leaves. If the male's smaller than her, he runs away. But if he's about the same size, and he just takes it, then she'll accept him. And that's one reason why they all seem to be the same size in the hole, because the same size pairing. And this makes sense because even though crabs grow at different rates depending on their food, then you've got a mate who's kind of your own age, right? So this is kind of interesting how that works out. The larger claw, in the hole, there's always a better spot, as usual, the most comfy couch, where you can get better food in, because there's water currents going by and you're filter feeding away. And there's usually a place in the rock crevice where you have a better water circulation. And the female uses her chelae to push and rock the male into that spot. And so she occupies the spot much more often and filter feeds much more often, but she needs to because she's producing the eggs. Eggs are very expensive. They're very big. They're full of proteins and RNA and all this good stuff. And sperm are very cheap. You can put thousands of them, with very little energy. And so the consensus in the animal kingdom is, eggs are expensive, sperm is cheap, and you have to invest in that way. Female rocks him out to get her optimum setting there. Doesn't seem to matter, lefties, righties, just a random mix. But strange crabs that show up at that hole are attacked. So they recognize strangers. So what happens when you take the mate away? When you take the mate away from the female, you take the male away, she sits in your hole. You take the female away, he gets out and runs around the aquarium like, oh my God, what's going on? So they take away the mate for different periods of time, 1 day, 2 days, 3 days, 4 days. After 2 days, the male do not recognize the female mate and they will arrest it. Even if it was their partner. Strange crabs versus their past mate get the same treatment after 2 days. That's the males. The females recognize the mate even after 10 days. So it's very interesting, these differences. Now, why? What's the advantage to that? That's lots of interesting questions, which are very hard to answer. Right? So that's that crab. Yeah, you never would have thought that tiny little crab had so much to offer us in the way of thinking about evolution and reproduction and what goes on there in these hidden worlds. So let's go to the beach. All right, this is Myers Creek, one of my favorite places. And I walk the rack line quite often. The rack line is simply where the surf washes up and brings stuff up. And probably all of you walk the beach and see this rack line. Well, some days there's a whole lot of little tiny jellies in the rack line. And Dave and Diane often send me photographs of their jellies. And if you get enough of them and you pick them home and put them in a dish of seawater, they will often be alive. This is a typical jelly. Yeah, probably how many people have seen one of these, the big moon jellies on the beach, yeah? And we're not going to talk about that one. But we are going to talk about their life cycles because that's important. So jellyfish are part of a bigger group called the Nigerians. They have stinging cells. They're not very complicated. They don't have a brain, for instance. But they have a very interesting life cycle. They have a dimorphic life cycle. One form is the jellyfish that you see. And one form is a polyp form like a sea anemone. Sea anemones are also part of this group. The sea anemones have often lost the medusa form, which is what the jellyfish is called. And the jellyfish has often lost the polyp form. But many, like if you were to go down to the marina at Charleston in the summertime, and you turn over the floats that have been floating in there for a long time, there's this white kind of jelly and goo stuff. You put the float underwater, those stromboli will float up in that white stuff, and you'll see the polyp form of the moon jelly. Now again, it's really tiny, right? You wouldn't know it. It just looks like goo when you turn it over. If you float, a lot of times putting things in a dish totally changes your attitude. So the polyp stage will bud off. It just clones itself into medusa. The medusa is sexual, male and female. So you have a sexual, asexual cycle. And seaweeds have this also, as well as some other invertebrates. So jellies hydrozoans have both stages still in general. Medusa are the jellyfish form. And the medusa can be found in the wrack, and they're usually very small. The hydrozoans have both polyp and medusa stages, right? And the polyps attached to rocks and floats and all kinds of stuff like that. So here's a typical medusa polyp. Here's a typical polyp of a hydrozoan from the, this is from the boat docks at Coos Bay. And this is very, very small. I don't know. The head's probably as big as the head of a tack. Right? But fantastic, beautiful creature. This is a medusa you find in the wreck line a lot. Put it in a dish, a lot of times it's still alive. And I always throw them back in the water, but it's a lost cause. There's thousands of them on the beach, you know. And this one here, I found just a couple times, but they're very different shapes and everything. So that's the kind of jelly you see. Here's the hydrozoans. Here's the polyp form of the hydrozoan. And these are literally 2 millimeters tall. This is on the leaf of the seaweed. You can see that there's a crisscrossing of this crisscross is the connection between the polyps. They're all connected with each other. These are the containers that the medusa get budded off of. So incredible life cycles and very odd structures you can find as you're out there in the intertidal zone. This is what the little polyps look like close up. So here's little tentacles. Here's the long tube. We're all connected together. This is their food tube, so they share food. All right. So they have stinging cells. They catch little bits of stuff in the water. They're filter feeders in a way. Predatory, kind of. So those are called the gonotheca. Gona, gonad. All right. You just kind of get the drift. And the little medusa bud off inside there. They're cloning. But weirdly enough, they clone themselves, but the clone doesn't look anything like them. It looks like a little jellyfish. What is a little jellyfish? So this is a completely different kind of jelly. These are the salps. Now, how many people have found something like that on the beach? And they're quite big, aren't they? So the salps are one of the most ubiquitous life forms in the ocean. They form chains of thousands and thousands of these. And it's a big part of the biomass. And things eat them. They're filter feeders. Water goes in, it's filtered by a gill basket, goes out. And so there's a significant amount of carbon cycling when in the ocean, because when they die, they float to the bottom, that kind of thing. Here is them up close. Again, they're not that complicated. You have here's the gill filter. Water goes in, through the gill filter, out. The food is moved down through food grooves into the guts and viscera. You know, it's not that, yes.
You said carbon cycling?
Right, so for instance, if you have a whole bunch of stuff that's dying, it sinks to the bottom, right? And if you're out in the open ocean, it sinks all the way to the bottom. And so the deep ocean depends on this organic litter for its food input. There's no photosynthesis going on down there. And so the cells, first of all, thousands and thousands are in these chains, and when they die, they sink. But also lots of organisms eat them. And so the carbon cycle is simply how does carbon move through the ecosystem? It's in the air, carbon dioxide is in the air. It's fixed by primary production by photosynthesis with algae and plankton and seaweed and stuff like that, or trees in our terrestrial case. And then that's eaten, turns into carbon in a fish or a tiny little amphipod. That's eaten. And so the carbon, but then when it dies, the carbon then sinks or gets consumed by bacteria and eventually turned back into carbon dioxide. So that carbon cycle is what drives the whole ecosystem when it comes to energy and food. And we're a big part of that, obviously. Everything is. Removing large parts of the carbon cycle is bad. So I was walking on the beach one day, and I found this very odd thing. I found this little barrel, which was clearly a salp tunic. But inside of it, there wasn't the salp. There was this really odd little crustacean thing that looks like an alien one. And weirdly enough, this is the people who saw this used it to model alien in Sigourney Weaver's movies. Right. So again, we are famous and no one knows it because invertebrates are basically the best alien thing around, right? So this is one eye. That's the eye. That whole thing. You can see how it's got maybe lots of little segments. This particular thing, this amphipod, eats out the cell and then uses the barrel to live in. And the barrel can maintain itself through absorbing organic material from the water. That's pretty wild. All right. And I had no knowledge of this. All right.
So the barrel is still alive?
Yeah. It has no brain. You know, it's not, there's nothing, there's no guts. So I got it home and I didn't know what this was. I just knew it had to be some kind of, you know, obviously arthropod. And you can see that the barrel, you can see this is the millimeter scale, right? And you can see this amazing claw with the little dots. The little dots are photoactive. There's a huge amount of biology in this thing that nobody knows about. So if you get closer, you can see the mouth parts, the giant eye, you can see the segmentation. It turns out that the female eats out the barrel, the female amphipod eats out the barrel of the cell, so just the barrel's there, and then she broods her babies in there. And she releases them and they hang out with her for a while. So pretty neat. You get it up close. This is under the microscope. You can see the eye much better, and those lovely dots. There's something about those dots that there's a kind of bacteria they think that is in the dots that produces a certain amount of bioluminescence. So one of the things that's interesting, of course, about this whole system is that they're transparent, right? So why are you transparent if you float around in the ocean? Why is that a good idea? Nobody can see you, right? Yeah, important. So this whole story of parasitism, people wonder, is this animal, this amphipod, is it a parasite or a predator? No, I'm not sure why we care because frankly, we know what it does. I consider it a predator because the cell is gone, just the barrel's left. But the barrel is alive and a parasite lives off a host. So what the heck, you know? So that's a question for a good discussion in class. So another kind of jelly are the pteropods. And these are the snails that have evolved to swim in the ocean and float in the ocean. They through evolution, their shell is severely reduced or gone. They make up a big amount of biomass in the living pelagic zone. Pelagic zone is the open ocean. Right. And the coastal areas where you get the primary production and lots of good plankton and stuff like that. So they're pelagic snails. This is like the poster child one, lives in the Arctic. It produces huge amounts of them and everything eats them. This one here is a serious predator. That one's the biggest. It's about this long. I've only found it a few times. It has these incredible jaws that it reaches out and chews up everything much bigger than it. They're abundant, they're important. And ocean acidification is a big problem for these because their larvae still have a shell. And the shell dissolves at a higher pH, lower pH. So as our ocean becomes more acid, this particular group of organisms is much more at risk than some of the others. So the some of them are mucus net feeders, where they produce this huge, giant mucus net, 2 meters across, little tiny creature. And the mucus net collects all the particles and stuff in the water, and then they eat it. Right? Some of them are predators, and lots of things eat them: turtles, tuna, salmon, birds. All other pteropods eat them, so they prey on each other. Here's one that I found in the rack line down at Myers Creek. It is a predatory pteropod. It's so cute. You know, you think, oh, it's kind of a little elephant-looking creature. It is really vicious, right? The appearances are not everything. So you can kind of see the sizes I found in the intertidal zone. These are the guts. There's no, I don't care about the cookies. Okay. Unless they're from Ben. So these are the guts in here. The shell is only in the larvae. And I've got a little video. My videos don't seem to work as well. You can kind of see wings. Now, one of the things that we don't think about because we're large, we're actually really gigantic organisms on the planet, is that these organisms don't live in water. Water is very, very sticky. So if you're this small, it's actually incredibly hard to move around in water. So the little wings kind of work, but you know, it's not like it's gonna go scooting booting through like a tuna fish or something. And you can see up close, there's these little great little nubs on the front. You can see the spots of the mantle. So a really lovely creature. And there were thousands of these in the rack line that day. So this is a predator. Here is its prey of choice. The prey of choice produces this giant, one of the big giant mucus nets. And when this creature senses that mucus net, touches it, it will extend something called a proboscis. And it will pull in the mucus net and until it gets to the creature. And then it takes these amazing jaws and tongues that have little horrible knife-looking things on it and cuts it out of its shell and eats it. So you see this little creature here, that little tiny creature? I opened it up, and this giant thing exploded out of it. That's the pharynx. The pharynx can extend way beyond its body length. It's all rolled up inside the body. And then when it senses that mucus net, it attaches it and can go way out and pull it in. Now, at the bottom of this pharynx are the jaws. And that mess right there. And if you're careful, you can dissect it out. Proboscis, here's the radula, here's where it goes into the tummy. And this is what the radula looks like. Look at that. That is evil. Now, all mollusks have this ribbon-like tongue covered with teeth called the radula. But radulas are different for each species, and the teeth are specific for the job they do. For instance, chitons, many chitons have iron magnetite on their radula because they scrape off rocks. They scrape algae off rocks, and it's a lot of work, and so their teeth are iron tipped. In this case, these are designed to kill and extract these poor unsuspecting mucus speakers. All right, so the last one we're going to talk about is the cucumbers. And sea cucumbers, you know, you pick it up. They're orange, they're black, they're clear, but they're often under rocks. You don't see them very often unless you really work at it. Sometimes you'll see the gill basket sticking out, but you won't actually see much of the body. So one of the things I want you to look for is that body has these things right here. What are those? What are all those things sticking out? They look kind of tentacly, don't they? I'll give you a hint. These are related to starfish. So if you saw those on a starfish, what would you call them? 2 feet. Okay. So cucumbers have 2 feet, just like because they're in the same group as the starfish. 2 feet. And I'll show you what they look like. Yeah. So tube, as in T-U-B-E. Look, I can use my whiteboard. Yeah. T-U-B-E. 2 feet. Like, yeah. I'm sure that everybody can see that way back there. 2 feet. I'll spell bigger. 2. How's that? There we go. And again, you probably can't see because it's dark. Oh well.
I'm going to turn the light on.
Okay. So yeah, it's all right. We got it. So who are cucumbers? Where do you find them and what do they do, right? Let's look at this. So first of all, sea cucumbers are echinoderms. Echino, spine, derm, dermatitis, dermatologist, skin, spiny skin, right? So once you know Latin, which I wish I had taken, but I never did, scientific terms become actually fairly easy. So the asteroid, aster star, the sea stars, right? There's some of our local species, the blood star, can we see it? The sand dollars, the echinoid, and that can also be the sea urchins. The crinoids. We have a local crinoid, but it's deep for sea. You'll never see it on the rack line or anything. but they're famous in the tropics, the sea otters. They're really amazing. They're very odd. They have this funky thing down here that grips the base, and they can turn upside down and swim around, and they do it. They're filter feeders. They're very odd, prehistoric. The ophoroids, the brittle stars, and the holothuroids. This is a local one we have here. The squares are 2 by 2 millimeters, so that's kind of a smaller species. I love the orange tentacle zone. So what do they all share in common? Why are they all in the Echinodermata? First of all, they all have pentaradial symmetry. Penta, 5. So they have 5 sides. And you can see that real clearly here in the brittle star 1, 3, 4, 5. A little less obvious in the sea cucumber, but the sea urchin, but if you look carefully, how many lines of tube feet are there? 1, 2, 3, 4, 5. Sand dollar, that's pretty obvious. And then there are 5 lines of tube feet in the sea cucumbers. So they have pentaradial symmetry, and there's more than I'm telling you about echinoderms. They're crazy, but one of the more unusual things is they have the water vascular system. So they don't use muscles to move their tube feet. And here's a close-up of the tube feet of a starfish. They have a whole water hydraulic system inside their body, like in a plane to lower and raise the wheels. They pull water in through this pore, as the starfish do, a pore at the top on their aboral surface. Water goes in, it goes down these channels. There's little bulbs that they can squeeze to push water into the tube feet. It's a whole hydraulic system. All of that kind of happens. Crazy. Right. They also all have some kind of hardened body part called the test in general, which is underneath the skin and helps support their body and protect them and do all that stuff. So you've seen this sea urchin kind of a thing, right? Mm-hmm. That's the test. The skin coats it. The little nubs are where the spines go, right? Sand dollar, these two are in the same group. In the ophoroids, all of these plates are all calcium carbonate. And the sea cucumbers look soft on the outside. You can't really feel those plates because they're little tiny rounded bits. They've evolved to kind of lose that main test. And now they simply just have a bunch of this kind of thing called ossicles underneath their skin. Right? So the sea cucumbers, their skeletons reduced to ossicles. They have feeding tentacles for suspension. Suspension means that they're doing this, the water's coming out. And deposit feeder means that this guy's vacuuming up, right? And they can be benthic, which means they live on the floor. They burrow, they live under rocks and crevices. One can swim in the deep sea. So one of the weirdest things about these guys is that they have feeding tentacles on this side. And if you remember the worm, he had the gills out there too with feeding tentacles, right? Sea urchins breathe through their butt. They bring water in through their anus into this respiratory trees. And that's how they get their oxygen. They can also get oxygen through their skin and through their tentacles. So they have this very unusual way of breathing. And here you can see kind of a depiction of this. Here's the respiratory trees. Water goes in, filters out. Okay. Oxygen can enter through the tube feet as well. So very unusual organisms. Here where we live, we have filter feeders. This is our local orange. Anybody ever see this one? Yeah. Diane's favorite. And parasitic puss, which is another big one. If you go to the CMLC, you'll see one of these in the touch tanks. This is one of the vacuum cleaners. Right. So here we have, this is a Hawaiian filter feeder. I mean, this is a Hawaiian deposit feeder, and it looks much better on my camera, but you can see it's got all these little feet out here, and I moved the rock because it's in the way. My hand. And then you see all those tentacles. So all day, for 10 to 15 hours a day, the sea cucumber wanders around and sticks to all kinds of little particles and puts them in his mouth. So that's a deposit feeder. Really wonderful to watch it in action. Now, what do sea cucumbers do in our ecosystem? Now, of course, much of it we don't know because we're humans and we can't possibly understand the complexity of the connections between organisms. But we've got some things down about these. First of all, they can be really, really a lot of them. So on one reef, for instance, there's one every 5 meters squared. Here in one tide pool, which is 1 meter, I've got 1, 2, 3, 4 in 1 square meter. Lots of cucumbers. In one reef in one small, tiny little reef in Australia, there were 2 million cucumbers. Lots of them. Well, so you saw that sea cucumber doing this, right? Well, and lots of them do vacuuming too, that kind of thing. And this moves sediment around. And why is that good? Well, first of all, there's a lot of bacteria in that sediment. And the bacteria quickly use up the oxygen. If we're going to have the mud flats, there's that lovely little section of the flat at the top of it, which is kind of grayish, brownish, and sandy looking. And then the bottom is this black mud. And it smells really bad, right? That's the anaerobic mud. That's where oxygen doesn't reach. And if you've ever clammed and you cut through the clam tube, you'll notice that the black mud is on the outside of the tube. The tube is lined with that gray, whitish color of mud. That's because that area, the clam is bringing oxygen into the burrow, and that's oxygenating the sediment. Well, if you want organisms to do well that aren't black, nasty bacteria, then you need this oxygen to penetrate into the sediment. And the sea cucumbers do that. They process the sediment, they mix up the earth, they eat a lot of the bacteria that are in and on that sediment. And they maintain the healthier microalgae bacteria layer where there's more oxygen. So a cucumber, the cucumbers are one area of this same reef, the Australian reef. In 1 year, you had 10,590 kilograms processed in 1,000 square meters per year. That's a lot of sediment. So they're really doing a good job. And they every 2.5 years, they rework the whole basin of the lagoon. So that's really important. We'll get to some of the results of that. Feeding. Well, they eat a bunch of decomposing stuff, which of course rots and takes away oxygen. So rotting process is one reason you get eutrophication in ponds. You know, the algae dies and goes down, the bacteria rots it and uses all oxygen, and then the fish die because there's no oxygen. So when you have the cucumbers eating all this rotting stuff, then of course it's not using up the oxygen in the water. They also excrete a lot, put out a lot of poo. And this produces nitrogen phosphates in the water for the growth of seaweeds and things like that, eelgrass. And they produce dissolved organic matter, which is actually absorbed by a lot of organisms for food as well. That's just organics in the water that can absorb through their skin. So if you want a healthy seagrass meadow, you need a lot of sea cucumbers. Seagrass meadows are one of the most highly productive environments on the planet. Humans use them for a huge amount of food and benefits. And in that seagrass meadow are millions of sea cucumbers, busy bioturbating, getting that oxygen down to the roots of the seagrass, getting rid of the bacteria.
Chlorophyll in there.
Well, there's chlorophyll in the seagrass, yeah, not in the cucumbers. This is a classic scene from our intertidal zone, which sadly is very rare now because of the urchin population scourge, which I'm not going to talk about today, but I have to tell you that it makes me very sad. But our seaweeds benefit a huge amount too because of the nutrients put into the water. So that's the ecosystem services of a sea cucumber, which I bet you remember. So but our sea cucumbers are actually endangered. They form a huge amount of protein and animal tissue that's eaten around the world. Now, you wouldn't think that since we don't personally eat sea cucumbers here, but in the Asian and Indonesia markets, there is a huge amount of sea cucumbers that are eaten in the process. This is a market in Indonesia. Those are all sea cucumbers. Okay. There you go. So these tend to be non-regulated fisheries, but with small boats, and they'll go out and they'll, collect sea cucumbers, sell them. So in 2012, Palau opened their market to exports to China. And in 48 days, 1,200 tons of sea cucumbers were exported from Palau, which believe me is not a very big place. The sea cucumber population took a real dive. Palau shut down the harvest. It was only 48 days long. And here we are in 2024, the population is still not recovered. They don't recover well. We do not want to end up with no sea cucumbers. It's one of those bricks, important brick. But you can see here in Sri Lanka, this is from 2010 data. This is just 2007, 400 million. I don't know what LKR means. It's probably a unit in Indonesia I don't know about, but there's a lot of sea cucumbers. The Galapagos, there's a lot of poaching because Galapagos is a protected reserve, and if sea cucumber poachers come at night, it's a tremendous problem. So we want to protect our sea cucumbers, and a lot of companies, a lot of countries have put in conservation things.
Can they be raised commercially?
They are. They're starting to farm them. And that's the way to go. I mean, that's the way to go with all this stuff. We cannot sustainably harvest wild populations for anything, including, for instance, our salmon. We want to eat a lot of salmon, we got to raise it. If you go to Europe, all the salmon in the markets, and there's a lot, is all commercially raised. I'm not a big fan, but the alternative is very little protein for 9 billion people. What are you going to do, right? If you harvest your wild populations, then they're gone, and then you don't have anything. So it's a big problem with how do we feed this many people? And as you guys know, you read the news, you're up on all that probably. There's a lot of great solutions out there, and people are working really hard at it. So now, this is the last thing, the most fun. Sea cucumbers look pretty defenseless, they're these little kind of sausage-looking soft things, right? Well, they have a lot of nasty chemicals in them, but most organisms get their way around that, especially with fish. So I'm in Hawaii, and I'm looking at this really cool, cute little sea cucumber here, this little brown guy with the black dots. I'm thinking, oh, how cute is that? And then suddenly there was this white sticky stuff all over my camera. It was like, where did all that come from? All right. And so it turns out that sea cucumbers can expel sticky stuff from their butt. And it's called the tubules of Cuvier. And believe me, if you're a fish and you get that thing stuck on your face, you could not open your mouth. So again, I have a little video which may or may not really work, but let's see how that works. So this is, I have a little sea cucumber abuse here because I have to poke it. So here's your, unsuspecting sea cucumber. Here comes Nancy's finger. I'm saying, come on, buddy, do your thing. I only have to do this once, right? Wow. And then I had to go and take it off with a toothbrush, and my camera. And what happens is sometimes they do that, and sometimes they just can't be bothered. I don't know the difference. Anyway, that's really fun. And yeah, I kept saying, "Can you give me some more here?" He said, "No, my batteries are out." I shot my wad. So here's another one. This one is here. I've used 3 sea cucumbers. All right, so these are a different species in Hawaii. I just call it the brown bumpy sea cucumber. It has a scientific name, which I've forgotten. And so then I go, okay, let's abuse that one. Oh my God, more comes out. Okay, and then there's a third one in here somewhere. Yeah, this one's a little more reluctant. And there. Oh my. And that stuff is eaten by the predator, or it sticks the predator's face together, and then this sea cucumber can go off and, live another day. What I'm not showing you, because I don't have any good videos of it, is that sea cucumbers can also do something called self-eviscerate. They can literally throw up all their guts. So the sea cucumber will, if it's really, here on our coast, the sea cucumber is a big parasiticus. If you bother it enough, it'll literally spit it insides out, the guts, the whole thing, everything. And there's just this tunic left. Predator eats the guts, and the sea cucumber will regrow all of its insides. Wow. Now that's amazing, right? So I'm just telling you that the amazing organisms in our intertidal zone, in our ocean, are so diverse and they have so many different ways of surviving and reproducing. Evolution has produced incredible diversity. And it's just really fun to share it with you. And I hope that you can enjoy looking it up yourself. These are RCP covers in Oregon. There's probably more. I just don't have photographs of them. The naked ones are really amazing. These live in the sand, kind of like clams, and they can burrow really fast. Most of the time when I try to get them out, I just end up with the cut off top, so I don't dig for them very often. It's very depressing. And then this one here, the Leptosynapta, is only about the size of my little finger, maybe half my little finger. But these brood their babies. So the babies mature inside the body wall. And then when they're mature, they just bust through it. They just make a hole in the parent. Yeah. So yeah, like, oh my God, you know, can't you like this one slightly different with that? But no. So just lots of different ways of reproducing and surviving in all these different organisms. So thank you. And I'll take questions. Yeah.
All of us in band, and I'm sure have seen the Valella.
The Valella, yeah.
And I've looked them up, and they're colonial.
Right.
And I'm having a hard time envisioning how different polyps make the
Oh, okay. So the whiteboard. Hopefully you can see. The Valella has. I had a math teacher whose name was Mr. Wright. You gotta love it. He was always right. He was the mother of man. And he taught himself to write left-handed, so he would never stand in front of the board with the students. I've never achieved this, ability. Anyway, so you have the sail. You have this is one polyp here. The whole sail is one polyp. Then you've got little, what look like tentacles coming down, right? Each of those is a polyp. Each of those is a zooid. And they have different jobs. Some of the zooids are going to be defense with little stinging cells. Some of the zooids will be ones that feed. And I think, Dave, is there a central zooid? A big central zooid? Right. So the structure of the colony is very complex inside. The Portuguese man of war is another one that is like. Yeah. Anybody else? Yeah.
I see on the beach the jellies that are rectangular or square.
Are they kind of just clear?
Yeah.
Those are pieces of cells.
Those are pieces of cells.
On occasion, you get lucky, you find a whole cell. Often that's gone. And cells, there's two kinds of cells that come up on our beach because they have a sexual and asexual stage, just like a lot of these organisms. So one of the stages is colonial, and the polyps are pretty big, and that's usually what we find.
Because you see them stacked.
Yeah, and then the other ones, the sexual ones, I think are pretty small. And so, but the ones you're finding are probably pieces of cells. I mean, we have a really common one here, which I showed you up here on the thing, but there's other species. Yeah.
So we got a little glimpse into the loss of biodiversity because of humans harvesting the sea cucumbers. Is there anything that we are not doing correct? Like I know we're doing ocean trawling. Yeah.
Oh,
trawling.
Yeah. Bad.
Do you have any comments on this?
The less we do, the better things are. You brought up trawling. I guess the thing that I can say to you, which is my personal kind of crusade, is that the commercial shell industry, where you find all those pretty little seashells, and you go into like Moe's and there's these cute little plastic covered bowls of shells, and then you go somewhere and there's these curtains of shells hanging down, or the I don't know, there are things you hang like the wind chime shells. All those shells are massive trawls scraped to the bottom of the ocean. They are brought up in lots of bycatch comes with them. Everything on the bottom of the ocean goes into those trawls and everything dies. So the starfish and all that stuff is thrown. And the shells get taken to, generally we're talking about third world countries. India is a big producer, Indonesia, the Philippines, which are not third world, but there's lots of places. And they are brought up and put in these massive yards, big piles of dead rotting mollusks. And then these fairly underpaid people are paid to, to remove all the biofilm, because a lot of these shells are infested with stuff. So you sit there all day on your little mat, cleaning off stinking shells. And a lot of them are broken, so they're thrown away. And the few that are good, they put in big bins and they sell them. So the amount of destruction occurs so that you can have a curtain of shells hanging is inconceivable to me. And yet we do it. So and when I was young, I did this. I bought shells. I had a big shell collection when I was young. I loved going to the shell shop and getting my tibias and all. It was just lovely. And then I found out when I was older about how harvest was done. And it made me really ill. So now I will buy, you can go to shell shops and ask for hand collected shells. Most people who are conchologists, in other words, they might be an engineer, but they collect shells for hobbies. People do this. They go to Australia and collect shells, but they'll do it by hand and they have a permit. That's all cool. You know, so, but I pick up shells on the beach. I love that. So that's just one example of an industry which is just decorative. But I've seen people in our intertidal zone. We used to have this lovely starfish here called Simpsoni, Solastro Simpsoni. It's the one on the picture of the physics book about the Woo Master. I don't know if you've ever seen it. It's like the Law of Physics, and there's this beautiful starfish. Well, that's ours. Well, it used to be ours. And I was out in the intertidal zone, I think it was 2006. And I found a few of these, which they're fairly rare in the intertidal. Now they're none anymore. And somebody was bringing a whole bucket of them up the beach. And I asked the person what they were going to do with them. He said, I'm going to dry them on my roof and put them in my art pieces. And I just said something like, well, you know, these are really rare. They're very old. They don't reproduce very fast, you know. Could you maybe think about putting most of them back? And the person did, which was very nice of them. But I doubt I would get that reaction from most people. I would say educating people on the value of looking and photographing and leaving is a really great way to think about our environment. You know, we're collectors, we're hunters, humans, I mean, we do that, you know. But the way you can hunt is you can find it, right? You don't have to take it. And we have this tendency, I mean, you really want to do that, but leaving it there, what are you going to do, put it on your shelf? And I was in the Galapagos this February. You can't take anything. You can't take a rock off it. And you have to go with a guide. There's only 16 people in a group. You can't take a stick. You can't go to the bathroom on that. I mean, they're really protected. There's a permit for everything you do. And I was taking pictures, and I realized there was no reason to take anything back. What am I going to do? Put it on my shelf and never look at it? But I can look at my pictures all the time. And so I think that we have this lovely gadget called phone, you know, and we have digital now. And I think that it really has changed our ability to enjoy nature without collecting. So that's just my little lecture. Sorry.
I would add to that, Steve has been involved with making a social plan in garden, and we're gonna have signage down there explaining to people about beach etiquette, what you should, you know, don't step in the tide pools, don't step on the rock, honor the animals down there, and don't take anything. And that's part of that education, is trying to get people to realize the fragility and the amazing animals that live there, and how they need to be protected. And it's a little part of the beach.
It is. I mean, one of the things I do as a scientist, you know, I know that these organisms are not at all, you know, they may or may not, they have decision-making processes. Most of it, in the case of these invertebrates, it's all chemical. You know, it's not like the sea. The sea cucumber decides to go one direction or another, right? It has a reaction to a chemical. But when I talk about the animals when I'm with my students, I have a lot of fun anthropomorphizing them. You know, I give them names. I've named all my whales, for instance, and I really love them for what they're doing, and I have fun tying them up. And if you do that with students, you find that they're much more protective of them because they have a name, because they're and you talk, you know, like I took a bunch of hermits to the 6th grade, which was kind of a terrifying experience. but the kids were really good because I talked about, you know, get to know your hermit. Give them a name, you know, Eli, Susan, whatever. And you know, what's it doing? What are those little antennae up to? And then I talk about how if you're scared, it'll freeze and die. That made a big impression because the arthropods do that. They'll do this. And they sometimes, if they're really scared, they just won't ever break out of that. I don't exactly know what it was. The frog picture was in my high school class. Stood there for 2 hours and then died. So, we had this one, and so I find anthropomorphic, chatter, and again, you covet it by, of course, you know, the hermit crab can care less what I think it's really doing. But it's awfully fun to do, and it creates a feeling of ownership, that this is an organism that, you know, has a life, which it does. So that is one way to do it. Even my seaweed, I love my seaweed. You know, and the students have started naming some of their seaweeds. It's like, yeah.
Do any of these animals have eyes?
Oh, yeah. There's photoreceptors. Obviously, this one has eyes, the crab. I do not know if the pteropods, I think they can sense light and dark. Dave, do they have photoreceptors? Dave and I have talked them before. Yeah, they have photoreceptors. Are they in the skin here? Or is, yeah. As far as I know, the sea cucumbers do not. The worms, this particular worm doesn't, but lots of worms do, and some polychaetes have actual real eyes. Lots of them have real eyes, especially the predator ones. And they have maybe 5 or 6 eyes.
They can spot something optically and react some way.
We don't know what their visual, we don't know what they see. This is kind of like a honeybee who sees a ultraviolet, right? We don't actually, our perception of that object may not at all be what the honeybee sees. When it comes to organisms like this, we don't know anything about the detail of their visual systems. I think they've studied octopus a lot. We have an eye, basically, this evolved separately, but very much anatomically like our own eye. Some of these just have eye spots, like the chambered nautilus that goes up, you know, the cool curly thing that's so famous. It has eye spots where it senses light and dark, but it doesn't form visual images. Now, when it comes to the polychaetes, I don't know if anybody's tried to figure out if they can see detail or just light and dark, but some of them have 10 eyes, big ones and then 5 little ones. So there's definitely a big component of visualization in some of these organisms. For instance, nudibranchs, the sea slugs, have eye spots where they can sense light and dark, but they can't see anything. So what cracks me up about the nudibranchs is they're absolutely beautiful. They have all these great colors, all these great forms, and they can't see anything. they can smell each other. They follow everything's chemosensory with them. So the visual, what we experience is not at all what they hear. Isn't that weird? It's pretty weird. Yeah.
In a university, in a healthcare setting, we had something like bench to bedside, taking the bench science and seeing how it affects bedside.
Mm-hmm.
Is that happening in your world with what you're seeing in the field and how it affects people personally?
Yeah, there's a whole, okay, I just read an interesting article, and this is kind of off topic in a way, but not. The old Palau urchin, debacle, sea cucumber debacle. Palau is a pretty small place. And so when they saw sea cucumbers, they shut down 48 days, that's it. But what was interesting is the article, one of the articles I read about this was focusing on the social implications. And what was true is that, and there's a lot of that going on, especially in areas where you have, well, like Charleston, where you have individual fishermen making a living, small boats, small fishermen, I don't know what they call it. They call them like, it's not artisan fishery, it's small boat fishery, whatever. And what's true is that it was examining the role of men and women in that fishery. Who's allowed to fish? Who does it affect? So it turns out that the men in Palau generally have the knowledge and the boat, and they're allowed to go out and fish. But then when the harvest comes in, the women are very affected by the processing. And so it looked at how the communities responded to both the excess export and then the crunching down of that fishery. And so there is a lot of research going on. For instance, marine reserve, which is another thing I heard in a lecture, I think, or maybe I read about. There was a study done where there were marine reserves in 2 different countries, very close together, somewhere in Indonesia or Java, somewhere. And one of it was an oligarch country, which was essentially fascism. You know, it was big cheeses in charge of everything, no democracy. The other one was a democratic country. And they tried, the company tried to get reserves started in both countries. In the democratic country, the village that's next to the reserve, they all met because it's democratic, and they all explored the values of this marine reserve. The whole group, the whole coastal communities were involved in deciding, and then they self-regulated. Because I mean, what are you gonna do? You're going out in the middle of nowhere. There's not like a police boat out there, believe me. So this group, that country, on purpose, the fishermen and the people harvesting control themselves. They chose to follow the rules. The other country was a top-down decision, and it didn't work because there was a huge amount of much more poaching. There was still poaching. It's not like everybody's nice over these other countries. But the point is that there was buy-in, a local buy-in that was agreed upon. Whereas over here, it was a command, and the local people will not consider that impact on the economy. So there's lots of research going on. Is that kind of what you asked?
Yeah, taking what you're learning, and if we totally kill this off, then you're not going to have the, we're not going to be able to develop the medicine that might save your child from whatever disease.
Yeah, I mean, it's the whole tropical rainforest story, right? You know, you cut down a piece of Amazon, you killed off 4, species that only live there, and what kind of medicine could they have produced? I think there's a movie with Sean Connery. So, anyway, yeah, I think that when it comes to, I think one of the reasons that I brought up the worm was because people tend to think worm-looking things are not that fun. They're ugly. Like lamprey, we got rid of our lamprey, right? And now what have we done to our salmon and our sea lions? The whole, whoa, that was a brick. Should be back in. And we're working on that. I think we're reintroducing lamprey. We're encouraging them. So we're doing, I think that there's a lot of good stuff going on. Yeah, wait, good question. Yeah.
So what's the etiquette for going out and exploring these tide pools? Like, it's okay to turn the rocks over and look
good question.
Stuff, and like what can you do?
So this is one thing I do. Most importantly, you don't hurt yourself. It's very slippery out there. I always have what I call controlled falling behavior, where I'm always working on 3 points of contact. Every step counts. And sometimes you have to step right on the sea urchin or right on something. These organisms are extremely tough, the ones that you're going to walk on, because they hit the pounding of the surf, and the surf is hitting them huge amounts of force. So in general, you're not going to do much damage to that upper. If you've ever stepped on a turban snail, it doesn't make much of an impression on a turban snail, except. But when you turn over the rock, all of the organisms on the bottom of the rock live there for a reason. They're on the bottom. You want to turn over the rock, and when you do, all the crabs will run anywhere. And I spend a lot of time moving crabs out of the way when I put the rock back so that then they'll get smushed. And then I turn over the rock, I look at it. If I take something and put it in my dish, I put it right back on the rock, and I usually get it a little wet, and I wait until it's stuck back. I mean, because these organisms live in microbial environments. Like nudibranchs, they're not really great at finding the food sources. I have this one nudibranch with red, it eats the red sponge. The tide was coming in, I took it out the rock, I took a picture, and I couldn't get to its sponge. So I put it near a red sponge, right? I go back 2 weeks later, it's still there in that same place. I'm going like, can't you move 4 inches, buddy? So I put him on his red sponge that time. So you have to be aware that these organisms live in these microhabitats. Moving is very risky. They're very tiny. If you have to go to another boulder, you can get eaten, you can get washed away. So if you start thinking on that scale, then you realize that you need to replace things back where they are. Then you turn the rock back over and go pick up another one, So yeah, that was a good question. Thank you for asking that.
I would like to point out for those of you who live locally, a great place to explore, the Tidepool, Cape Blanco.
Oh yeah.
Right north. I was with school language, field trips.
Fun.
I think the trail's still there. I looked down, it looks like it's kind of steep up there.
Oh no, I've been, yeah.
But you've been there.
Oh yeah. North.
On low tides.
That's a lovely place.
That's the best place I've ever seen.
Good.
And there's lots of it. It's not like Sunset Bay or anything. There's lots of it. That goes, it's a lot.
No,
the whole thing.
Blanco's real, and Blanco's pretty accessible.
There's whole ships out there who are real deep. I mean, they say.
Is that where
That's the beginning of Port Oakland.
Right.
Beginning of Port Oakland.
I think well, Rocky Point's a good spot.
Yeah.
I haven't, Humbug, I've been to Humbug, there's some interesting seaweed there.
So have any oysters down there?
Not at Rocky.
Oh.
Sorry, they have, plants, but a lot of people plant rock.
Oh.
So anyway, Yeah.
Language.
Language is good.
Made it through fifth grade till we had to move. That's like called Will Code.
Yeah.
Wilbur.
Anyway, thank you very much.